So let's say you found a skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
All right, hello everyone, and welcome to the Sunflower versus Dr. Dan Stern Cardinal debate. We are debating today. Let me make sure I get this right. Viral evolution falsifies the strong, irreducible complexity argument. Dr. Dan is taking the affirmative. Sunflower is taking the negative on this question. And by way of introduction, Dr. Dan is, of course, a professor at Rutgers University where he teaches various biology classes. And he did his PhD thesis on, in fact, the effects of evolution on viruses. So he is the only person I would ask to talk about this subject that I know. And of course, we have Sunflower, who frequents modern day debate and other discussion fora about this type of topic. And so we are going to be doing about maximum 12-minute openings. Both of my speakers have assured me that they are very unlikely to actually take up the entire 12 minutes, but I'll be timing them anyway. Then we're going to go to an open discussion until the top of the hour, at which point we will open it up to a Q&A. Apparently my audio is low. It is a little low. Why is that? That's weird. Well, I'm putting it up right now. Let me see if I can't fix that real quick while I continue to talk to you all. Let's see. So, super chats will be collected automatically. And so if you do have a question and you really, really want it answered, that is the most likely. But let's see. Oh, there I am, loud again. Sorry about that. For some reason, OBS decided that my mic isn't the mic it wanted to use again. And it just was like, oh, did you want me to use your Oculus Rift mic? Which is, the answer is no. No, I don't want that, OBS. Thank you. Anyway, so without any further ado, I'm going to turn it over to Sunflower for his presentation.
I'd be happy if I learned something, and I'm willing to, concede that if I learned something. Oh, sure, no problem, no problem. Okay, so hi everyone. I'm Sunflower. Some of you might know me, some might not. I just want to say thank you so much to Dapper Dino for having us, and thank you so much to Dr. Dan for taking the time to talk. I will be, of course, defending my position, and I think I have a good position, but I'm also not here to win cheap gotchas or try to, do some kind of, unfair characterization of either side. If I learn something, I think that's actually good, and I think I probably will. So with that said, I'll go into my, what I have to offer here. So, all right, I'm going to go ahead and share my screen, and that's going to start with a clip from a talk that Dr. Dan had with Michael Behe. So tell me if this is showing up.
That is showing up.
Okay, so it's just going to start off with a question.
You better not strike my channel with this.
Don't worry, he won't. So Dr. Dan is basically asking, how would you falsify irreducible complexity? And this is the answer Behe gives.
Evolution of which we have not specifically documented, and say, well, that's the thing that can't evolve. So let's get that out of the way first. How do we falsify this idea like any other scientific?
Well, first off, to falsify it, yes, you could show, Darwinists could show that something that he or she thought was irreducibly complex had indeed evolved by random mutation and natural selection. Say a two-protein system that both proteins.
I'm going to stop right there, and I want everyone to keep in mind what Behe just did there. He said a two-protein system, and he held up both of his this to kind of put them together, right? And so this is what I kind of want to hone in on for a second. Let me go ahead and try to share a different tab now. I click this and then click screen. Now I'll just show the whole window. Yes. Okay. All right. So the reason I'm referring to this is because it seems to me that when we're talking about systems, the bare minimum for a system in a biological setting would, or in a biochemical setting, would probably involve at least 2 proteins. Now, Dr. Dan's example here that he's sort of led with is 1 protein. Okay, it's the VPU protein of the human, HIV variant. And while there are indeed at least 4 specific point mutations, these are all still 4 specific point mutations that change, the conformation or what have you of this protein that allow it to antagonize the tethering. So I just want to start sort of open here with saying, like, is a single protein considered a system? I don't really know. I don't really think that's a great characterization of a system in this context. I guess it could be argued that it is, but I kind of, that was one thing that jumped out at me, and I feel like Behe's sort of in opening with that definitely aligned with my intuitive understanding of what the bare minimum of a system would be considered. So to move on from there, I want to introduce something called the quasi-species model, which is essentially, this is a simplified explanation, right? When evolutionary biologists describe competition between species, they generally assume that each species is a single genotype whose descendants are mostly accurate copies. This was to be calling, high fidelity, high reproductive fidelity, so on and so forth. And then, some organisms or genotypes may exist in circumstances of low fidelity, where most descendants contain one mutation. So I'm bringing. Oh, sure. I can zoom in. Yeah, let me see. How is that? Is that a little bit better? Okay, so essentially this concept of the quasi-species model is a model in which most descendants contain one or more mutations, and the group of the genotypes is constantly changing. So discussions of which single genotype is the most fit become meaningless. Now, this is often applied to viruses. Viruses, especially RNA viruses, extremely readily fit this concept of a quasi-species. And so I think that it's important to bring this up in the concept of this argument of this topic, and how things, especially of viral evolution versus biological evolution, since viruses are not living, and the fact that the quasi-species is often referenced, and what that really, all that really means is that there's so many genotypes that are all having mutations that no single one of these is even considered to be kind of like the original specimen whose genome is being replicated and fitness analyzed. It's like they exist in like a cloud, so to speak. So yeah, survival of the flattest. I thought that was interesting. It's so in quasi-species, it's not necessarily survival of the fittest, survival of the flattest, which has to do with the cloud that these exist in. Okay. So moving on from there, there are unique properties in viruses, and especially how they evolve, that don't apply to other biological organisms.
You may want to zoom in again.
Oh, okay, sure, yeah. So this is from a paper, if anyone cares. It's called Viral Fitness Definitions, Measurement, and Current Insights from 2012. I'm just using really one part of it, but yeah, people can look if they want. So essentially, there's unique properties to viruses in that they have two kinds of fitness, right? They have what would be called replicative fitness within a given host. And then they have a different kind of fitness, which is sort of being newly examined, which would be like inter host, like the ability to jump from host to host. So these are two different sort of concepts with viral fitness that don't really apply to other things. And now what I thought was interesting is that there's a fish virus in which the virus that jumped from species to a new species, right? So the virus that had jumped to rainbow trout as a new host had actually lost fitness in the previous host, which was sockeye salmon. So this raises an interesting question. If a virus gains maybe potentially a novel function in that it gains the ability to jump to a new kind of host, but then it loses fitness in the previous host, and let's say that the new host species begins to die or begins to develop immunity to it, can that really be seen as a like a gain of function, or that seems more like a detriment to the virus, depending on how you're defining it? I mean, it becomes more fit in its ability to replicate in the new species, but if the new species rapidly dies and it's now unfit for the old species, well, then if the virus rapidly goes out of existence, was that really a good mutation for the virus? I would say no. I mean, because what landscape we were working with here, because viruses are so unique, you can analyze a virus and say it's got great fitness within this one species, or you could even go beyond that and say within this one person or thing within this species. But if it can't, is that a fit virus or not a fit virus if it can't jump from a species to another? Questions like this, which I think are just very very interesting. Oh yeah, I think this is actually just expanding on that. Fitness landscapes of intermediate viruses. Again, this is a paper called Role of Evolutionary Intermediates in the Host Adaptation of Cane. Oh yeah, this is an expansion on the previous one. Let me see. Oh, I lost my place. Oh yeah, so basically it's pointing out that there's significant fitness values that would have clearly limited the number of viable evolutionary pathways that could have followed and carried, blah blah blah. So what it's saying is like with viruses, it's not even clear where a function or an ability to shift hosts is this limiting or expanding the functionality of the virus? Again, that's kind of what I'm going at here. This is just very just showing that I think I already went here actually. Yeah, it's one protein with 4 mutations. Oh sorry, am I running low on time? I'm back. 2.5 minutes. Okay. The last thing I'll bring up is this. So HIV-1 groups M and N emerged in the last century following 2 independent cross species. So this was like there was actually 2 groups that went from chimpanzees to humans. So M is the one that is the pandemic that we all deal with in human civilization. But there's also one called N, which are exceedingly rare, with only about a dozen infections identified ever. I might not have time to fully go into this here, but what's interesting is that this N group had also the same mutation that oh sorry, I might have to zoom in. The N group still antagonized human tethering. But there's other aspects of it that make it so that it's just been absolutely borderline non-existent compared to the M as in Mary group, which is a pandemic that's obviously the one everyone knows. So I just thought that was interesting that it might not only be the ability of the protein to antagonize tethering. There's other aspects of what made this group proliferate. And with that, I will conclude my opening. So thanks so much.
All right, you had 50 seconds left. So good job on that one. Dan, I will start the clock on your first go. But if you want to first share your screen or whatever, we will put that up. And then once you're ready, I will start the clock. Dan's working on it. He's muted, but he's working on it. So in the meantime, I do want to remind everyone, please do hit the like button. It really helps.
You think I would have had PowerPoint in the full screen view ready to go, but no.
All right, I'm starting.
Dapper, can you just switch it back to where the faces are adjacent, if you don't mind? Thank you. Perfect. So, hi everybody. If you don't know me, I don't know why you're watching this if you don't know me, because I feel like that would be weird that there'd not be overlap there. But if you don't, hi, I'm Dan. I'm an evolutionary biologist, and I'm thrilled to be talking about viral evolution. This is kind of my thing. So thank you, Dapper, for hosting. Thank you, Sunflower, for participating. I love the topics you touched on in your intro. These are just, I'm so excited for this chat now. This is going to be great. So we're talking about irreducible complexity, which is a concept proposed by Dr. Michael Behe. He's a biochemist at Lehigh University. He kind of proposed this idea in Darwin's Black Box in 1996, and I'm going to quote at length just to make sure that we have the specific definition of irreducible complexity. He defines it as a single system which is necessarily composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. An irreducibly complex system cannot be produced directly, that is, by continuously improving the initial function, which continues to work the same mechanisms by slight successive modifications of a precursor system, because any precursor to an irreducibly complex system that is missing a part is, by definition, non-functional. I apologize, but there is more to this, because he continues. An irreducibly complex biological system, if there is such a thing, would be a powerful challenge to Darwinian evolution. Since natural selection can only choose systems that are already working, then if a biological system cannot be produced gradually, it would have to arise as an integrated unit in one fell swoop for natural selection to have anything to act on. That second paragraph on this slide is really important, because the first one is just like defining how you identify an irreducibly complex system. This one is the important part. This is the the hypothesis of irreducible complexity. And basically, the hypothesis is if a system is irreducibly complex, it's a problem for evolution. It's a challenge to evolve that system. Now, it's not that simple, because there's a bunch of different ways we can interpret this. And there's really three forms of the irreducibly complex argument. All three of them are invalid for different reasons. Now, the first one is you have to ask the question, does this hypothesis permit all evolutionary mechanisms? Behe is very specific in terms of describing, numerous successive small steps, same function through the whole process, right? He's describing very strict Darwinian evolution. So if you literally take that definition, the answer is no, you don't permit all the non-Darwinian evolutionary mechanisms in the context of IC. And if you interpret it that way, then irreducible complexity doesn't even address the question of can a system evolve? Because it's just omitting a bunch of evolutionary mechanisms. Now, if we put that aside, we say we're not going to be a strict textualist here. We're going to say he obviously means to make a general critique. He's not limiting it to the mechanisms of evolution that we knew of in 1860. So we'll say, okay, we permit all the mechanisms. Now the question is, are there systems that meet the criteria he described that can evolve? And there are two forms of the argument you can get from this question. If the answer is yes, this is the weak form of the irreducible complexity hypothesis, that, well, some systems that have irreducible complexity can evolve, and other systems absolutely 100% cannot. That is unfalsifiable, because that just becomes creationist whack-a-mole, right? It's, well, here's how this thing evolves. But what about this other thing? Well, here's how that could evolve. Oh, well, what about this other thing, right? It's never ending. The version of this question that we care about tonight is the other form, the strong form of this argument. It's, can some systems that have irreducible complexity evolve? No, absolutely not. 100% of systems that meet the criteria for irreducible complexity cannot evolve, right? That's the strong form of the argument, and that's what we're talking about tonight. And this form of the argument is false, because we have observed the evolution of irreducibly complex systems. We've done it in viruses, we've done it in bacteria, we've done it in eukaryotes. For tonight, we're going to ignore those other things, and we're just going to talk about viruses. So the counterexamples here are pretty straightforward, and there's like a whole bunch of them. Basically, if you take the strong form of this argument that nothing that has irreducible complexity can evolve, then the observation of the evolution of any system that meets the criteria falsifies that hypothesis, and we can look at viruses and find examples that falsify the hypothesis. They check all the boxes in that definition of irreducible complexity. And I want to note, when I was chatting with Behe, I did the same thing and read those and said, is this like a good definition? Does this still hold? And he said, yep, still good. So that is a valid definition. When I asked Dr. Michael Behe about it back in March, I think that was. So the first counterexample is something we already heard about, a little protein called VPU in HIV. This is specifically HIV-1 group M VPU. It's the pandemic group of HIV. Now this right here, SIV-CPZ, that's simian immunodeficiency virus, CPZ for chimpanzee. It has one function. It messes with this protein called CD4. But the HIV-1 group M version does a second thing. It messes with this protein called tetherin. That's part of our immune system. It's different from the chimp version, so we had to find HIV had to find a new way to inactivate that protein. Well, it does it through VPU forming what's called a pentamer. So it's at least 4 mutations, right? So this is an irreducible trait because we've done the tests where it's in 2 regions of the protein. You need at least 3 mutations over here and then at least 1 in the other region. So it's at least 4, and you need them all in order to have this trait. And it's actually 2 binding sites on the VPU that matter. Well, it's actually 3 technically, because they form a ring, a pentamer, where basically you need a right hand and a left hand, and they hold hands in a circle. But then you also have to interact with the tetherin protein itself. So you have a bunch of different interactions going on. You could say it's just 1 protein because it's VPU, but really it's 3 binding sites affecting 2 proteins and 5 or 6 participants in that system. So that's HIV-1 VPU. That's an irreducible trait. It crossed into humans somewhere in the early 20th century. So like that's one example. Another example is in lambdaphage. This is a bacteriophage. And this is the ability of lambdaphage to use a novel receptor to get into its E. coli host. Because what E. coli will do when there are viruses around, you end up selecting for E. coli that just don't have the receptor. Well, the phages can actually evolve to use a different protein. So in this experiment, they again found, in this case, 4 mutations that are required for this. 3 wouldn't do it. And you can see right here, 3 of the lines that use the new receptor and the gray boxes indicate the mutations that are present. These 2 mutations right here, 1 and 2, those are not optional. You got to have both of those. And then the 2 on the right over here and the 2 on the left over here, you got to have 1 from each of those. So you could either have this one or this one, and you need to have either this one or this one. So you need 4 mutations. Any 3 of those in combination doesn't do it. You can't use the new receptor. You need to have all 4. That's irreducible complexity. It requires 4 simultaneous new biochemical traits, mutations, all at one time, without any selection for the intermediate state, without any gradual increase in functionality. Cool thing about this is these viruses, they can use the new receptor. They can also still use the old receptor. And in fact, some of them, and there's another table, another figure from this paper that's big and complicated, but it shows a bunch of the different sets of mutations that are involved here. And some of those actually, not only do they use a new receptor, but they're better than the ancestral state at using the old receptor. So that's another directly observed. This was in a laboratory experiment. It didn't take long to evolve. This is a directly observed example of an irreducibly complex trait evolving in viruses, which again, the strong form of the hypothesis is invalidated with one example of this type of trait evolving. So to summarize this argument, both of these cases involve traits that meet the criteria for irreducible complexity. It's not just me strawmanning what irreducible complexity is. It's criteria that Dr. Behe agreed on just within this calendar year. They were traits that were absent from the ancestral populations. Both SIV and the ancestral lambdaphage don't have these traits, so we know when they evolved. And they were directly observed evolving, meaning like in the lab, we did it and then we sequenced them and documented it, or we have the HIV pandemic and we have sequences going back to the 60s for the HIV. I think 1959, I think, is the earliest one we have sequence for. So we have the sequences and we can compare them to the ancestral SIV state, and we know when these viruses first appeared in humans, so we know the time frame where these changes occurred. Now, Dabber, how am I doing on time? Do I have another minute or two? 2 minutes, perfect. So the conclusion here is viral evolution is 100% sufficient to falsify the strong form of the irreducible complexity hypothesis. So I was taking notes during Sunflower's intro. I have a bunch of things here, but the big thing that I want to cover, alive viruses, alive, not alive, whatever. Alive isn't a useful thing. All these different things. The important thing for our discussion is that none of this stuff matters when it comes to irreducible complexity and can those traits evolve specifically in viruses. Right? None of the, measuring viral fitness, it is complicated. Quasi-species, really cool dynamics, but none of that stuff matters when it comes to can traits that we can define as irreducibly complex evolve. So that's where I'll end it. Thank you all for listening.
All right, well, you ended it with 1 minute and 11 seconds left to go. So what we are going to do now is we're going to do some open discussion. I would like Sunflower to start it off, just because he went, Dan went most recently, so I figured that Dan got everything he wanted to say out. And right now, I'm not going to be timing either speaker, but if there starts to be significant overtalk or crosstalk, I will start setting a timer. It'll probably be a 3-minute timer. So, guys, don't make me start a timer for you guys. So anyway, Sunflower, take it away.
Okay, sure. Thanks. That was a really great opening. Yeah, it was interesting. I was engaged the whole time. I guess my initial question would just be, so if the VPU protein, let's hypothetically, if it was only 1 point mutation that we pinpointed it, and we said it's 1 point mutation that makes this protein able to antagonize tetherin and thus infect humans, would that then change your understanding of it as being irreducibly complex?
Yeah, absolutely. That would make it not irreducibly complex because it would be just a single mutation. So it wouldn't be 2 or more that need to simultaneously be there. If it was just one, and hypothetically this one mutation, right, allowed it to make that ring in the same way that, like, for for hemoglobin, for example, right, with sickle cell disease, it's one single point mutation, one amino acid substitution that makes the hemoglobin go from the functional tetrameric protein to they form these long fibers, right? It's just they end up kind of fitting together. This mutation on one side of the protein, not only does it change the properties of that one side, it becomes kind of compatible with the other side, so they all stick together, right? That's not an irreducible trait because it's one change, but it facilitates both sides of that interaction, right? But VPU isn't like that.
Well, I guess that then, I don't know. For me, I just feel like the fact that 4 mutations are contributing to changes on one protein versus there's examples where you could see one mutation contributing to, like, the one you just explained, or maybe there's others way more complicated conformational changes in a protein. I feel like the fact that it's 4 mutations and then using that as 4 parts, that even though they're mutating one protein, this is where I'm having a little bit of trouble with defining, like, what the parts of the system are. Again, and I'm referencing Behe saying, like, the first thing he said when you asked him, how do I falsify this, was if you have 2 proteins mashing together. It seems like that would be the bare minimum for me. I don't know.
In this system, though, you have an interaction of 5 proteins. They're all VPU, but it's 5 individual proteins that are required for the system. So you need each one has to, it's the right hand and the left hand, and they're all holding hands in a circle, right? So you need 2 sets of changes, one to each side of the molecule, basically, and then they can link up in this big ring. Now, hypothetically, let's stick with this. Hypothetically, if that pentamer, instead of being formed by one VPU protein, right, just 5, like 5 of them, right? Let's say, what if it was a hexamer and it alternated between 2 proteins, one A, B, A, B, A, B in a ring, right? But the number of mutations was the same, the way they interacted was the same, the way they functioned was the same in terms of inactivating tethering. Would that, for you, cross the line then into making it an irreducible system?
Yes, that absolutely would, 100%.
But that's biochemically, that's a totally arbitrary distinction, because now you're taking a system that's actually functionally, like, it has the same number. I mean, it's 6. I made it 6 versus 5 to make like to make the numbers even. But like, let's pretend it was a hexamer instead of a pentamer of VPU, just to keep it a 1-to-1, because I think your argument holds regardless of the number of proteins in the ring, right? Is that a fair characterization?
Yes,
yeah.
Yes, it is.
So just to keep it 1-to-1, right? Let's pretend it's a hexamer instead of a pentamer of VPU, right? And it's all VPU versus alternating VPU and something else. What's the distinction there in terms of evolving that system? It's the same number of mutations. It's the same number of protein-protein interactions. The fact that it's using 1 protein for the ring versus 2 is arbitrary in terms of meeting Behe's criteria.
I would say because the VPU protein had previous functionality that was not in any way impacted by these mutations. So, like, for example, my understanding of the irreducibly complex argument is that what seems to be irreducibly complex would be synergistic as in at least 2 different proteins have a synergistic effect with their own mutations that are acting synergistically. So if a single protein can remain functional and accumulate accumulate mutations, and then eventually that same single protein sort of fits into a place it didn't usually fit before, I just don't see that as being this sort of bare minimum synergistic system-based, I guess, complexity or synergy that, if you will, that I would say is required for that definition.
My dog is barking to get in the room, so just give me a second.
No worries.
Okay. And while we're working on dog things, I want to point out that Gary DeVere, who's been a member for 3 months at the tetrapod level, said, as Covert Cuttlefish said, smash that like button. So please, if you are enjoying this, do hit the like button. It really does help a lot. So, sorry, Dan, go ahead.
Okay. So first thing with having multiple functions you kind of touched on at the beginning there, VPU does do another thing, right? It does that CD4 inactivation. And HIV1 group M VPU still does that. So it still messes with this completely other biochemical system, and it inactivates, it antagonizes the CD4 protein, but then acquires this other function. So this is what's called exaptation, where it starts off doing one thing, it now does another thing. Now that can be a couple of ways. One, it can just switch functions, right? Feathers, right, that went from thermal insulation to flight is an example of it switched functions. The wing feathers on a bird, those are specifically for flight. They're no longer for thermoregulation. Although I suppose maybe they are a little bit. I don't know enough about bird physiology to know how much the wing.
They thermoregulate eggs during brooding.
Right. Okay. So eggs during brooding. Okay. Bad example. See, I shouldn't have picked the dinosaur example when Dapper Dino was here. So you can come up with examples where it's a complete switch of function. You can come up, like some enzymes do that, where it did one thing and now it does a totally separate thing. This is not that. This is where it is a very straightforward gain of function, right? So that's the first thing. This is just, it did A, it still does A, but now it does B. But the more important thing is this disagreement over whether this is irreducibly complex, because does it have the required level of synergy among different components in the system? And the reason the answer is yes, it is irreducibly complex, is I'm just going to pull up that definition if I could find my PowerPoint window. There it is. It's let's see. Here's the exact wording from Behe. And again, I ran this by him and he was like, "Yep, that's good." So
if you're trying to share, it's not coming up.
I'm not sharing. I'm just going to read. It's so necessarily composed, right? So necessarily composed, it needs to have all the parts, right? Several well-matched interacting parts. Okay, the different amino acids involved in the right hand, left hand, plus the interacting with tethering. So composed of several well-matched interacting parts contribute to the basic function, tethering antagonism, wherein the removal of any one causes the system to cease functioning. So if any of those new mutations aren't there, it doesn't work. And in
Wait,
this case, there's at least
but it still works to do the CD4 stuff.
but it doesn't do. But my contention is specifically that the tethering antagonism, the novel trait of antagonizing tethering, is irreducibly complex. That trait specifically.
But that seems to not really that doesn't seem to map onto the definition. Then if it's a function that if it's a protein that functions for one thing and continues to function for that same thing, but then develops what you could see as a new function. I mean, I could see but couldn't. I feel like that's an arbitrary definition of a new function. Like, why are we defining this tethering ability to antagonize tethering as a function?
Because all of the viruses in this family, so the simian immunodeficiency viruses, and there's gorilla and African green monkey and pseudomangabe and chimpanzee and human, right? All these immunodeficiency viruses, they're in a group. I think they're I'm like 90% sure they're lentiviruses. I should know that 100% sure. I'm like 90% sure they're lentiviruses. And the way they infect their hosts, right, all these primates, is that they need to deal with this protein called tethering. And if you don't prevent tethering from operating, then you cannot infect your host. What tethering does is when the viruses are ready to leave the cell after they infect it, tethering, as you can guess from the name, actually grabs onto them and keeps them on the surface where they can be destroyed. So you can't infect if tethering is doing its job. Now, the other SIVs, they have various means of dealing with tethering. A lot of them use a protein called NEF, NEF. NEF destroys tethering via interacting with a part of the tethering protein that human tethering doesn't have. Our tetherins are actually smaller. They have like this little tail in most primates. Our tetherins don't. We have a deletion in them. That tail is missing. So this SIV protein NEF, it can't target tethering in humans the way it does in all these other species. So that's like tethering antagonism. It's a biochemical thing that these viruses do. It's a function of these viruses. It's something they must accomplish in order to infect their host. HIVs cannot do tethering antagonism the way their ancestors do it, right? SIV doesn't use VPU to do it. It uses NEF, right? I should say SIV CPZ doesn't use VPU. It uses NEF. So the use of VPU as the tethering antagonist, that is a novel biochemical trait. It's a new set of interactions between a viral protein and, in this case, itself, and also a human protein, where those interactions didn't previously exist. So those are novel biochemical traits that we find in both HIV1 group M, which is what we're talking about, but then you mentioned earlier group N, which does it a slightly different way, and it's not as good at it, but it still accomplishes the same basic thing.
Okay, so that does make sense. So what I gather from that is that essentially, if we define the functionality, a more of sort of a general functionality of the virus, as in the virus functions to infect its host, then we can introduce the novel functionality of VPU to antagonize tethering in humans. Then that would align with B's definition, because it would be to say the system is not just VPU. The system is the virus of which VPU is a part, and like there's an irreducibly complex component of the virus. That maybe I'm bastardizing the terminology there, but it does at least make more sense to me to view it more as VPU isn't gaining more functionality, strictly speaking, for what VPU is supposed to do, but the virus is gaining more functionality as it can now infect a new host with VPU.
I think it's fair to say VPU is directly gaining, like these mutations are, I think, pretty straightforward, meet the criteria for a gain of function mutations in VPU. But let's look at this a slightly different way. Consider this, right? There's a bunch of different ways for organisms to break down glucose, right? There's a bunch of different biochemical processes that you can use to break down glucose. So you can a lot of things, like everything does, like glycolysis. But then from there, you could do lactic acid fermentation, you could do ethanol fermentation, you could do pyruvate oxidation. Like, you've got a bunch of options after you do glycolysis. Now, let's say, hypothetically, you've got an organism that can't do, for whatever reason, it can't do pyruvate oxidation. Its ancestors did pyruvate oxidation. That's another step in the process, right? This cell, for whatever reason, it's mutated. It can't do pyruvate oxidation for whatever reason. But some other enzymes that it had are similar enough that it could maybe interact with the products of glycolysis, and it mutates, right? Just by chance. These are, mutations are probabilistic. It's not trying to do anything, but it's probabilistic. And a handful of mutations occur, and it can now do lactic acid fermentation. It could do a different metabolic process, right? That would count as a new trait, right? It's taking a target molecule and it's doing something with it that the ancestor could not do, right? It's a new biochemical thing that particular cell can do, right? That counts as a new trait, right?
Okay, yes, that's a good example. And in that case, I would call that a novel trait because it helps the organism survive and reproduce. But isn't that crucial? I mean, like, for example, there's a mutation that causes hemophilia, right? So like, but so then I guess, and I don't mean to like just sort of pivot, but like, would we call whatever leads to hemophilia in humans, which is like, like the most catastrophic thing you could ever have, you basically can't live. If you get a cut, you just bleed, you never clot. Would we just say that because a new thing is going on in the blood clotting, whatever, mechanism, a new thing is happening? Does every new thing that happens count as a gain of function, or does it have to actually increase the sort of, reproductive success overall of the thing?
It doesn't have to increase the reproductive success, but it has to increase like the sum total of biochemical reactions that are happening, right? So for hemophilia, that's actually a loss of function, because the way blood clotting works, it's a positive feedback loop that involves a bunch of like intermediate signaling steps. And most forms of hemophilia involve a deficiency to one of the signals in the middle. So you've got like factor 7 deficiency or factor 9 deficiency. And somewhere in that series of signals that would normally amplify and amplify and amplify, that you have a break in the chain and it stops. So the amplification doesn't happen and the clot doesn't form correctly, right? So that's a loss of function. It's not that it's doing something new. It's actually losing the previous function that existed, which is not what's happening in HIV.
Well, that's interesting though, because I generally, and I don't mean to, I'm not saying that you've ever done this, but I generally regard this kind of loss of function, gain of function distinction as kind of like iffy. I mean, like, don't usually, isn't it kind of a constant talking point when that creationists use when they sort of they'll refer to the genome of something and they'll say like, oh, like evolutionists are calling this a novel trait when really it was there all along, but they just like lost part of it. Isn't that kind of?
Yeah, that happens. So like an example where you hear that is like sickle cell, where you have with sickle cell, so a little bit of genetics for everybody, you've got 2 alleles for each gene, right? One from mom, one from dad. If they're different from each other, that's called heterozygous, right? If you're heterozygous for the sickle cell allele, like you got the mutant allele from dad, but you got the regular hemoglobin gene from mom, being in that state is actually not terrible because you're resistant to some kinds of malaria. Now, creationists argue, well, sure, that's beneficial in the context of your fitness if you're in an area with malaria, but it's actually a reduction in fitness if you talk about fitness in terms of functionality, because you have a broken allele, right? It's that some of the hemoglobin is mutated and that's interfering with the malaria, but like that mutated hemoglobin doesn't work. But again, that's not what we have here. BPU hasn't broken, it hasn't lost its ability to do its other functions. It's just gained this new biochemical function that facilitates infection and reproduction of a new host.
Right. Okay. Wait. I'm trying to think of the points you just made. So wait. So you're saying the reason hemophilia. So hemophilia is a clear loss of function genetically.
Yeah.
And so is sickle cell.
Sickle cell could go either way because it's really difficult, because you could argue, right? You can make an argument that so what's going on, the word for what's going on with sickle cell is called antagonistic pleiotropy. Pleiotropy is when one gene has multiple things going on and they have they are opposite effects on like benefiting your fitness, right? So like the sickle cell allele, right? It's bad because it could lead to sickle cell disease, but it's good because resistance to malaria. So that's antagonistic pleiotropy. It affects two phenotypes and they have opposite effects on fitness.
Okay,
so that's the word for that.
And so would it be fair to say like we only really know like when do we determine whether or not something is a bona fide gain of a trait, or like because if I lose a trait, like let's say hemophilia in some sort of weird scenario, and maybe there are examples of this, where if you like loss of functionality leads to better reproductive fitness. That just now counts. Well, yeah, and if that just now counts as reproductive fitness, then I don't see why it's useful to distinguish that hemophilia is a loss of function. Like, isn't it kind of like the result is more what we're focusing on here, not necessarily the nuts and bolts of the genetics that are being sort of dealt with, right?
Right. So there's two separate things here. There's like functionality and biochemical functions and the nuts and bolts, and then kind of separate to that is fitness. How does it affect the reproductive capacity of the individuals that have the trait? And when it comes to irreducible complexity, those are separate questions. And the definition of irreducible complexity is really about those biochemical nuts and bolts.
Okay.
How many interacting parts do you need to confer this new trait? It's I'm trying to think of an example. Here's an example for you. Here's an example of a trait that is more complexity, but could also have a fitness cost. And I would definitely consider it a gain of function. So there's types of antibiotic resistance that are called efflux pumps, where basically the bacterial cell, it'll be a mutation to a protein pump that takes waste products and pumps them out of the cell, right? Those pumps tend to be specific to whatever waste product it is. They can mutate to be able to grab onto antibiotics and pump those out of cells. Now, in some cases, those proteins can do double duty. They just kind of broaden their target so they can pump whatever waste product it is. And then there's an antibiotic that's pretty similar to it, and they can pump that out of the cell too. So the antibiotic doesn't work anymore. That's a gain of function. But here's the thing, that costs more energy to have that generalist pump because it's just operating more, right? It's doing more pumping. So now in certain contexts, it might actually be harmful to have that mutation because you're spending more energy compared to the genotypes without it. So that would be like a gain of function, right? Functionality goes up. You're doing a new biochemical thing that you didn't do before, but there is potentially a fitness cost to that. Does that make sense?
Yes, that makes a ton of sense. That actually, what you just described, definitely reframes things for me, because I guess what I was more or less I could even reframe my previous example. Like, if it were the case that hemophilia was not the result of a loss of genetic functionality, but it was, let's say, like there was actually some, I don't know, some new protein that like interacted in a novel way or something to this nature.
Yeah, something that interfered with that pathway, but it did something else useful, right?
What if it interfered with the pathway, but there was more there was more stuff going on? like, is that how we're defining? Because that's where I'm getting hung up.
Yeah, I mean,
like, if there's more stuff going on, period, in like more reactions, I guess, or energy expenditure, biochemical energy expenditure, does that itself, is that all you need to say something is a novel function?
it's yeah. So I'm gonna just pull up a picture I use in class. If just give me 1 second here to kind of pull this up. This will just take a second. And I'm going to share my screen because this gets right at this. Um, share, share screen, share this one. No, I don't want that. I want this. Here we go. Okay, so this is actually a slide I use in my intro to evolution class, talking about how you get new traits. And one of them is exactly the kind of mutations we're talking about, gain of function mutations. So you have a protein in blue here. This is a protein, and it interacts with some substrate, right? This protein mutates, and now it's got some other characteristic to its surface, and now it interacts with this other substrate. Now it's totally possible for that novel interaction to be two things at once. It could be both irreducible. Actually, let's call it three things at once, right? It's a gain of function because it's doing a new thing, right? It has this new thing that it's doing. Two, it could be irreducible if the number of mutations and the characteristics of that interaction are such that it checks, Behe's boxes, right? So it could be an irreducible trait. And as you're describing, this could be a harmful trait, right? Let's say this protein grabs onto, factor 9 or something in the blood clotting cascade. Well, that's not good, because now that factor 9 is going to be stuck to this protein instead of doing whatever signaling it's supposed to be doing. So what you're describing in just hypothetically can totally happen. But in terms of irreducible complexity, only one part of that is relevant, and it's the nature of that new trait. Does it have multiple parts that are required, yada, 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 right? That's the only thing that matters for the question of irreducible complexity.
Okay. So I don't know. I not but like, can you pull that bomb thing back up again?
Yeah, the picture. Yeah, I'll pull that back up. Yeah.
Yeah.
Yeah, it does look like a cartoon bomb that Wile E. Coyote would use, right?
Oh, was it not supposed to be a bomb?
No, it's just a protein, and I needed different shapes sticking off the side.
You gotta put Acme on it.
I should put Acme on it. That would be funny.
Sorry, I just thought, I didn't mean to
Yeah, there you go.
Okay. Anyway, so I'm the red arrow, the red chevron thing here would be that's the new. Yeah. Okay. I'm saying, what if that is something that is harmful to it's only harmful. There's no beneficial thing. It's just pure harm.
It could still be irreducibly complex, though, because irreducible complexity, nowhere in that definition does it talk about it being beneficial or whatever. It doesn't mention fitness. It's just describing the way the components of the system interact.
Well, yeah, but do you see how this is kind of to me, this feels like just a great I mean, okay, I'm not going to argue that you technically you're just going by the definition. What you're saying might be true, but I have a hard time feeling like this is a useful example. I just feel like if you could invoke any mutation at all, and as long as it's interacting in a unique way, involving, I guess, more than one part, but even if it's killing the organism, that's still irreducibly complex.
It's still irreducible. It's still irreducibly complex. Now, the nice thing about this hypothetical is that it doesn't matter. Isn't that nifty? The next slide. I knew the next slide was the exact thing. So I literally just took the slide from this class. So the nice thing about the VPU example is that none of those worries affect that example, because it isn't lethal, it isn't harmful, it doesn't have a downside for HIV. It's a new biochemical trait where, yeah, it does this new thing and it requires specific interactions to do it, but it also maintains this ancestral set of interactions. It still does the old thing. It didn't lose the old thing. The new thing is beneficial, not harmful. the fitness of the virus is increased by the presence of the new trait because it has to be. If it can't infect tether, if it can't antagonize tetherin, then it doesn't infect humans. You can knock out VPU, no infectivity. You could replace it with chimp VPU, no infectivity. So we have all these hypotheticals and where it gets like kind of gray. None of it matters for this example. This example is cut and dry.
Fair enough. Well, question. Can HIV group M, can that infect chimps?
That's a great question. I don't know.
Because
I don't know.
And I mean, I mean, you made a good point there with the fact that, like, I'm kind of like, I don't mean to like just, I feel like you make really informed points, then I kind of just bounce off them and bring up some kind of like sort of my own like just knee-jerk response. I don't mean to do that, but
that's okay.
Yeah, but so I'm thinking like, all right, hypothetical, and this actually will tie in my entire position together. Hypothetically, let's say the HIV-1 group M, this variant, it lets the HIV infect human via the antagonization of tetherin, and now it's a pandemic in humans. What if that then leads to humans, like, through whatever, whether it be developing natural resistance or developing some kind of like, antiviral treatment? If humans do that, and then this form of HIV, the HIV-1M, it proliferates, and then like all the chimps, the SIV variant effectively goes away. HIV-1 goes back into the chimps, and it's the only one available. But then humans create like a way to destroy HIV-1M, and then they eradicate it. And then if it had just remained simian SIV, it would have still been in existence. Right. You see where I'm going with this?
Yeah, like, I see. I see what you're saying. Yeah.
Is that a gain of function in the grander scheme of things?
So, I mean, if it can do a new biochemical function, yes, because that's a different question from its either fitness, like in the moment fitness, which is what natural selection can deal with, and it's the only thing natural selection can deal with, right? Natural selection can't like plan, hypotheticals. It's what works in the here and now. And it also, what was the second thing I was going to say? It was, okay, so the question was, yeah, it's still a gain of function because it's doing new biochemical things. It doesn't matter if it ultimately goes extinct in that situation. That doesn't have a bearing on did it come up with a new way to antagonize tetherin or not. Like, yes, it still did this new way to antagonize tetherin. Now, you could talk about, well, what effect did that have on the long-term viability of the population? Did that lead it to get into a new ecological niche that was unstable and then it goes extinct? Like, you could have all these knock-on discussions for that.
Right.
But none of that stuff is relevant to the question of characterizing that specific biochemical trait.
Okay.
So we are going to be wrapping up the Q&A. If you guys want, I will be open to giving you guys like a 2-minute closer if you want. If you don't, we'll just start with questions. So does either of you want a 2-minute or so closer?
I would. No. Honestly, I mean, I would give 30 seconds just to say like, thank you. Okay. All right. So 30 seconds.
30 seconds.
Yeah. Thank you again to Dapper Dino and to Dr. Dan. Again, I came in trying to be sort of with more of an open mind, and Dr. Dan really brought up some really good points that I was not aware of, and I'm just happy to say that I was able to learn some things, and I still have some very minor sort of, I guess, in my own mind, like difficulties with the understanding of what describes a function, but I will say that Dr. Dan not only addressed my points well, but also informed my understanding of these topics. And I feel grateful for that, and I really, I mean, I'm just happy I was able to have this discussion. So thanks.
All right, and Dan, do you want to have a quick closer?
I'll take 30 seconds. Say thank you for hosting, Sunflower. Thank you for kind of wanting to do this. This was super fun. I'm sure you can tell, and I'm sure everyone else can tell, that I just love talking about viruses. I can talk about viral evolution. This is just my thing. I'll talk about it all night long. Don't get me started. So this was super fun. I think this is great. And also, I love these conversations because, like, specifically conversations like this, where it's kind of questions and honing in on like where disagreements are. It's a really useful way to like get right to the heart of the issue. And as a teacher, it's actually really helpful for me because it helps me hone my way of explaining things in such a way that avoids or addresses where there's potentially confusion and misunderstanding. And that is, I always find that really helpful. So, so thank you all around. This was fun. I hope everybody enjoyed that conversation.
All right, thank you. And I have to say, both of you were very nice and polite, and the chat, I've had several messages about how this is one of the most civil debates that I've hosted, which I appreciate. Also, both of you have very long conceptions of what a second is.
Yeah, I have issues with perception of time.
No, don't worry about it.
I apologize for that. Yeah.
Dan went just as long as you did. You both took about a minute when you said 30 seconds.
30 seconds, yeah. No.
Okay, so we're going to start in on questions. So this is how this is going to work. First, super chats do jump to the top of the list. So if you definitely want your question answered, I mean, that's the safest way to go about it, but it's certainly not necessary. What's going to happen is, if a question is for both parties, I'm going to give them each 2 minutes to address the question. If it's for only one party, then the person who gets the question will get 3 minutes. Then the other person will get 1.5 minutes, followed by a final follow-up for up to 1 minute by the person it was for. Now, if any person wants to waive some of their time, that's fine. The next person doesn't get any less time unless they also waive it. To make sure that I notice your question, please tag @DapperDinosaur. Remember, the SAUR is now part of the channel name, so you are going to have to do that. If you're used to just typing Dapper Dino, it won't actually highlight it for me. And our first question is from Mr. Wilford. I can say words. For both, do you think a biological system which is deleterious in most environmental contexts could be irreducibly complex? And I do think we discussed this a bit, but since it's for both, and we had Dan go most recently, I'm going to have Sunflower go ahead.
So wait, tell me if I'm understanding. You said, could a biological system that's deleterious in most environments still be irreducibly complex? Was that the question?
Yes.
Well, yes. I think Dr. Dan actually demonstrated that. There's complexities to the what he really did was demonstrate that it's a separate question as to the ultimate sort of, I guess, I don't know if the fitness is the right word. The ultimate long-term survival of something is kind of a separate question as to whether or not there's short-term functionality in its ability to adapt to its immediate environment. So, so the fact that the question said most environments, not all, I don't know. I guess I would say it's possible. Yeah, I don't know.
Okay. And Dan, you may also have up to 2 minutes, although I think you probably don't need it since we did already discuss this quite a bit.
I think yes, it's possible.
All right. That is nice and concise. We have a $5 super chat, which isn't a question, but this is from Jordan K, who says, a reasonable discussion between two individuals on the internet? Impossible. Well, we're here to prove that is not impossible. It's just unlikely. Now, I do have a question that I did notice. However, it wasn't tagged, so I did almost miss it. This is from Gfish, so please remember, do try to tag me so that it, my eyeballs get drawn to it. This is for Dan, and it is, please define a gain of function. So Dan, you have up to 3 minutes starting now.
Yeah, gain of function. So it's funny because this is one of those fun things in bio where you think it's really simple. And then like it is kind of initially like, what's a gain of function? Oh, well, if it can do something new, then it's a gain of function. And then you look into it and it gets a lot more complicated than that. So like some specific examples of things that we would consider gain of function. If you have an enzyme that mutates and it has a new target that it can do whatever it does with, right? It has a new target, then that's a gain of function. It does something new. If you have a structure, well, I guess it wouldn't be a gain of function mutation or structure because there'll be multiple mutations. If you have a mutation that leads to the development of a novel structure, right? You could have a mutation to, a regulatory scheme during embryonic development, and maybe part of the body is different. You get an extra loop in the intestines or something, and now you can do something else with that. If you have a duplication, is a good example of a gain of function, right? You get a duplication. Now you have a gene or set of genes that did one thing, and then it duplicates. Oh, well, now you've two sets of genes that do a thing, and then subsequent mutations. You can have what's called neo-functionalization, where following that duplication, it could then have a new target, have a new metabolic pathway, whatever the case is. Those are all examples of gain of function mutations, but they don't have to be like that. You could also, in cancer, for example, you find lots of gain of function mutations where the cells get really good, for example, at inducing the growth of blood vessels into the cancerous region because they need more resources, oxygen, and they generate more waste. So they need more blood vessels. Well, that's a gain of function for that tumor. It's bad for you, right? It's bad for the person with it. But for those cells, that is a gain of function. You are increasing their ability to do whatever it is they're doing, right? So there's different ways of defining gain of function, and they don't all have to be beneficial for the organism.
Okay, and Sunflower, would you like the option to respond?
I'm not gonna have nearly as like that. Whatever Dr. Dan said, go by that. Yeah, I struggle with actually defining it, so. I don't have a great answer. Yeah.
Bio is complicated. Turns out it's really complicated.
Yeah, indeed.
All right. Well, this next one is a question for both parties, and this is from Griffin Hunter, who asks, this is a question for, okay, I already said that. Do you think that neuroevolution AI development, where the AI is integrated in generations, says anything about biological evolution? So we'll have Dan go ahead.
I don't know anything about like AI stuff. I know they do use, there's machine learning and evolutionary algorithms involved, but like I don't know anything about the nuts and bolts, so I'm gonna no comment that one.
Okay. And
I'm sorry. It said, what their question was, as in AI is integrated or implemented with generations.
So the idea here is, so neuroevolution is you're developing a neural network for an AI. So it has inputs and it has outputs, and it usually will have neurons in between. And then the strength of various inputs and how they interact with the intermediate neurons will then determine which output is resulted. And some people program these by just having a whole bunch of them perform tasks, setting a fitness function. The ones that do the best then mutate and they do the function again, and they'll iterate that several hundred times or several thousand times. And do you think that this, which is a thing that people do in computing, has any says anything about biological evolution?
Yeah, I'm gonna. So I have a tangential understanding of neural networks, and a somewhat understanding of biological systems. I'm sorry, that's question is out of the scope of my. I wish I could answer that. I feel like I'd probably get the Nobel or something, but I just don't know.
All right, and I did get confirmation from Griffin Hunter that is in fact what he was talking about. So, since we're currently waiting on a question, I'll say I do actually think that evolutionary algorithms do have something to say about evolution in biology. I do think, though, that we should be careful not to apply those findings too broadly, because the way that computers are coded, even with genetic algorithms, is very different than the way biology works. But there is something to be said for the fact that the neuroevolution type AI development can result in neural networks that humans cannot figure out, and yet they do perform the task. And I think that is something significant. So I'll just say that. Let's see. Edgelord 420 says, speaking of gain of function, anyone here ever heard about the gain of function research at Wuhan lab?
You know what? I actually looked into that briefly before this debate, hoping that it would be relevant, but to my, frustration, there doesn't. I was hoping I could like map that onto this, but I didn't really find much overlap. It seems like most of the at least as far as I looked, most of the people were just like, oh, there was a like a high level 4 virology lab in Wuhan, and it just so happened that it broke out there. What are the odds that that would have been an accident? But I didn't actually, I couldn't find anything relating to like the genes of the virus or anything like that. So,
And Dan, you can also take 2 minutes on that, up to 2 minutes.
It didn't come from the lab, people. Didn't come from the lab. Came from probably bats. Didn't come from a lab. We could do the phylogenetics. It's in
But
there.
what
with
if
the
they
bat
were
stuff.
laboratory bats?
If they were laboratory, it still came from the bats. It's so the lab conspiracy theories are like, oh, it was on purpose. Like, no, it came from the bats. The only way it came from a lab is if they isolated it and then someone got sloppy. Like, but that's it's not sinister. That's just sloppy lab work. But that probably didn't happen either. It just came from the
yeah,
bats.
and like I said, I was like looking for, I was like hoping that I would find like, I will look into this and I will find clear examples of human fingerprints on on viral genomes, and then I'll be able to reference that in my whole argument, and then I just didn't find anything. So I was like, oh.
It turns out the evidence for anything related to that is basically zero. Yeah.
All right.
Not good.
We have a question from Griffin Hunter, who says, "Do both?" It says competitors. I'm going to go with interlocutors. Think that Edgelord 420 was just being an edgelord or not? And I mean, his name is Edgelord 420.
I think yes, that was my assumption. Yeah.
Right.
Yes.
Yeah. It's person named Edgelord says Edgelord thing. I mean, it's kind of what you expect.
News at 11. Yeah.
Right. There was also a comment about genetic algorithms and pointing out that they are used in many things besides neural networking, which is very true. There are even vehicle designs that are done by a genetic algorithm where things like airplanes and automobiles are actually designed by mutating automobile and airplane designs. Okay, and for $5, Mr. Wilford, and I think this is for both because it doesn't specify, asks, but what if the pandemic came from the bat cave? Shocked emoji. Does anyone have...
This is my give me a break face.
Okay. Sunflower?
I'm legally forbidden from commenting on that.
Oh.
So yeah, I actually can't comment on that. Sorry.
Well, I'm just going to point out that it could have come from the Batcave, but the best part of the Batcave is, of course, the T-Rex, because the Batcave has a T-Rex. In case you didn't know, it doesn't in any of the movies, but it does in the comics. Yeah. That's why the comics are obviously better, because they have more T-Rex than Batman. So, so G-Fish makes a comment, and I don't actually know the truth value of this, so I would like to ask if anyone here does. So he says Wuhan is not where COVID-19 began, and I have no idea to what degree that's true or false. Does anyone actually have any information on that?
I think in terms of the region, that is where it began.
I mean, why post that comment without like saying where he thinks it did? You know, it did or
came from the moon. It came from the moon. This is actually the Andromeda strain. It came from space.
Well, I would say I do know that the first cases were reported in Wuhan, although I guess it's possible that they were in some other neighboring region and they just weren't reported.
Yeah, tell me more. Whoever made that comment, like, I want to hear more. Where are you going with this? Like,
historical thing with that. The first reported cases of HIV were not in Central Africa, but once we started widely surveying and figuring out like at the time you couldn't do widespread genome sequencing because it was the early 80s, but like once we started, figuring it out, there were reports of diseases that fit that profile going back through the 70s in parts of Central Africa. So even before we could do the genome sequencing, we were pretty certain we knew where HIV came from. And then once you get up into the 2000s, now you can do widespread genome sequencing and piece together the history of it. And it's very clear that it came from Central Africa, right? That was where the initial crossover into humans happened. The nice thing about 2020 and 2021, is that we could do all that sequencing in real time, right? We did this also in 2009 with the swine flu pandemic. We were able to figure out where it came from in real time. And so in terms of regionally, we can't say like the first instance of COVID-19 that occurred in any person anywhere in the world was in Wuhan. But what we can say is that the most recent common ancestor of all of the documented cases was there, which strongly implies that it started there. Right? That's what we can say based on the data we have. And by Wuhan, I mean the region, not the lab
Or the city.
or the city. Right? I'm talking regionally.
There's a
Yeah,
whole lot
and
of
I
like
agree with
nested
that.
bits. There's there's the province, there's the city, then there's various locations in the city. Yeah.
It's all good, G-Fish.
Yeah, no. Let's see. Oh, there we go. Iron Charioteer points out the virus came from melting the permafrost. China has a lot of people generating heat. There you go. It's an ancient virus from the permafrost. Except that we've noticed. Correct me if I'm wrong, but the reason that we know it came from bats is we actually found a like sister lineage in bats, and we can actually look at things like fixation rates in the two populations and work back how long ago it came about, and it's
Exactly.
recent.
If you do the phylogeny like this, it's like bat, SARS coronavirus 2, bat, except it's like 50.
Right.
It's just all bat, SARS coronavirus 1, bat, SARS coronavirus 2, bat. It's all bat coronaviruses. So like,
all right,
where it came
now
from.
Nefereti says, for sunflower, what do you think about evolution through artificial selection? For example, agriculture, like broccoli.
I mean, is there any way you could ask them like to be more specific? Like when someone says, "What do you think about?"
Okay.
Well, I accept it as a thing. It like I like broccoli.
Well, let me see if I can clarify, and Nefereti can let me know if I do a good job.
Sure, yeah.
In agriculture, especially in many cases in our food stocks, so things like in the case of broccoli, its ancestor is a wild mustard plant. We've turned it into broccoli, as well as things used for mustard and kale and cauliflower, and there's like 8 different vegetables.
Oh yeah, cauliflower, kohlrabi, Brussels sprouts, cabbage, broccoli, and you're selecting for different parts of the plant to be big, but it's all the same ancestral plant.
Does the degree to which humans can induce gross morphological changes in organisms that we use in terms of agriculture say anything about the possibility that nature could do the same in wild organisms?
Okay, that's a very interesting question. I mean, with that question, I guess what I think about when I hear that question is that there's like when usually people think about the word environment, we tend to think about sort of the inanimate, or not inanimate, but like sort of the like
abiotic.
Yeah, sure. The abiotic, or maybe the abiotic and or the inanimate biotic, sort of like the circumstances upon which we dwell. When in reality, is the environment kind of more of in the case of human agriculture, something that's kind of shaping things in a way that you might not see if you're only zoomed in on one aspect. So I guess what the question is like the same way that the thing we call just like the capital E environment overall of the Earth, is it possible that the effect of human agriculture has a sort of similar grand but hard to notice gradual effect on the shaping of things, that sort of like the just the general thing that we might consider like, oh, it's just the weather, or like the cycle of this, like maybe like these things are comparable. I think that's interesting, but I think that I unless you invoke an intelligent agent for the capital E environment, like the weather and the Earth and the cosmos, and like I think there is a meaningful distinction to be at least made, even if groups of intelligent humans you can predict patterns. I still think I would have to hear an argument for what the intelligent thing behind the kind of like big cosmic environment is that's responsible. But I don't know. Maybe it provoked too much thought in me. Let's get on to the next question before I ramble too much.
Well, first, Dr. Dan does have an opportunity to give some kind of response from it. No? Okay. All right,
no, I think I mean, artificial. I will say, if you read Origin of Species by Charles Darwin, 1859, like the first quarter of the book is all about artificial selection, selective breeding. Like he's basically saying, look, we do it. It's just matching your environment does the same thing. If you do better in your environment, the process works the same. He's very like upfront of like, look, we all agree on this one thing. I'm just arguing it also happens over here. That's kind of how he frames his argument, which is cool.
Okay. Well, our next question is from Silver Harlow, who super chatted in to jump ahead in line. And Silver Harlow says, Sunflower, has this discussion helped you understand why, quote, irreducible complexity is not something regular biologists think about?
With all due respect, that I sort of see that as a condescending question. Look, I came into this with an open mind. The way the question is framed is to me that sounds like, in so many words, have you admitted that you were nowhere near as informed as you thought you were? It's like, okay, well, look, I'm willing to accept that I've learned things, but if you're gonna, I don't have a lot of interest in questions like that, to be quite frank.
All right, Dan, do you have anything you want to add?
Oh, I mean, I think that's perfectly fair.
Okay. So let's see. The next question that we have is from Fernando Katakai, which I think the X on the end of that name is silent, and I'm going with that. This is a question for Dr. Dan. Why do bats carry so many viruses?
Oh, that's a fun question. So the answer is that everything carries so many viruses. We carry a ton of viruses. You're full of viruses right now. It's kind of frightening. We're just infected with viruses all the time. Most of them don't really cause us trouble. So for example, there's a whole class of viruses called cytomegaloviruses that, like, most people by adulthood have gotten cytomegaloviruses. They just fly around. You generally get them. Certainly by middle age, almost everyone's gotten them. And I read one estimate one time of like kind of where energy is spent in our immune system, and by one estimate, if you've had cytomegalovirus or are infected by cytomegaloviruses, they tend to be persistent infections. What this one study, and I don't think this is a consensus, but it's just to give you an idea of the scope, is that 10% of the energy of our immune system is spent just dealing with this one group of viruses, because they're just everywhere and we're constantly getting infected and we're constantly fighting them off. And unless you're immunocompromised, we win easily. They don't make you sick. There's no symptoms. It's fine. But like, they're just around and we constantly have to deal with them. We're full of tons of viruses all the time. The average human gets four colds a year. I forget I think it's 3 something. It's in the low to mid single digits colds per year, right? And there's like dozens of viruses, hundreds possibly, that cause a common cold. We just get viruses all the time. It's, bats get viruses, sure. So do us, and horses and birds and everything else gets tons of viruses, and they're always flying back and forth and infecting each other. It's not just bats; they're not special. Everyone gets lots of viruses.
All right, I do want to say, Iron Charioteer says, hug your local bat. Do not do that. Bats can carry rabies.
No, yeah, don't do that. Don't do that. That's possibly the virus with the lowest survival rate that we know of is rabies. The number of cases of survival and recovery from rabies, and I'm using the word recovery fairly loosely there, but like you can count them on 2 hands.
Yeah. And they result in severe neurological damage, lifelong.
Like if you don't get the treatment, right? If you don't get the rabies shot after a potential exposure, and you actually get rabies, the survival rate is essentially 0. Mm-hmm. It's don't.
And your prognosis is that if you survive, which is extremely unlikely, you will be mentally handicapped for the rest of your life.
Mess with you forever. Don't get rabies.
You'll also have your physical capacities diminished because your coordination will go down, your ability to think will go down. It'll ruin your life. Don't hug bats.
Don't hug bats. Or raccoons. Don't hug raccoons either.
Nope, don't do that.
Don't do it. Don't do it.
In fact, if you're going to come in close contact with either of these animals, make it be one that's actually captive and you're a professional in caring for it. Otherwise, just stay away from wild animals, basically.
Just don't do it.
Yeah.
Just don't hug wild animals. It's a good rule.
The real question, though, is can whales carry rabies?
I don't think so.
I don't know of any confirmed cases, but actually we do. So, Sunflower, if you do want to talk about bats, you can have a minute and a half to talk about bats.
No, I don't have. I don't really have anything to say about that. Bats are weird.
Okay, so I just Googled. I love how we're not in left field. We're not even in the stadium anymore. But I just Googled, can whales carry rabies? And the first response from a website called whalefacts.org that exists. Whalefacts.org.
Good to know.
Believe it or not, whales can acquire rabies. However, the likelihood of a whale catching rabies is immensely low.
Okay. All right. Well, speaking of whales and doing things you might not think of, though, I did learn at one point that one of the major predators of moose is the orca. Because apparently, you might not know this, but moose like to eat seaweed. So they'll swim into fairly deep water, and moose dive. So, yeah, apparently diving moose occasionally get preyed upon by orcas. Let's see. This is a question for both from Fernando Katakai, who says, do viruses compete with each other? And I'll start with Dan, since he already seems to have an enthusiastic answer.
Yeah, they do. Viruses compete with each other all the time, and it's so cool. This is why sometimes a line of influenza, for example, might go extinct, because a different line of influenza, which uses the same host, right, our respiratory systems, comes along and is a better competitor in that environment. So this new flu comes along, it's a better competitor, and you maybe don't have a pre-existing immunity to it because it's never existed before. You've never gotten infected by it before. It comes along, it's a better competitor, and it drives the earlier strain of influenza to extinction. That's a thing that happens. It's so cool. Yeah, viruses compete all the time.
All right, and Sunflower, do you have anything you want to add?
Yeah, that was kind of. Well, I feel like maybe I meant to touch on that kind of in my thing, in my debate proposal, and it's like if we look at the quasi-species thing, and we try to apply the conventional kind of definition of fitness and function and whatnot. It's like, well, we have a original viral substrate or whatever that infects the host, and then if one of its progeny is this competing virus that eradicates the original. Well, now it's like you have an example where the original viral line had a mutation that was beneficial that also made the previous version extinct.
Oh, yeah. Happens all the time. Yep. I mean, this is what we're seeing with COVID, right? Where we have these different variants, and basically in the context of a pandemic, the selective pressures, and Sunflower, this is exactly what you touched on in your opening when you talked about intra versus inter-host competition, right? Where you have to be good at like winning in the host, but you also have to be able to get to a new host. And depending on the ecological context, like how many susceptible hosts are there, right? So like at the beginning of a pandemic, everyone's susceptible. But after a bajillion people have been either infected or vaccinated, relatively few people are susceptible. Well, now the selective pressures affecting that virus are going to be different. So certain mutations can occur that lead to new variants that are going to be selected for in that new ecological context. We're seeing this right now with SARS coronavirus 2. We saw it in the early 20th century with the Spanish flu in 1918 and 1919. That virus persisted into the 1950s, but it only killed 50 to 100 million people in that first season, right? 1918 into 1919. After that, it wasn't this super deadly influenza anymore. Why not? Because it was operating under different selective pressures that made it less dangerous, right? So yeah, viruses are constantly competing with each other. You're getting new mutations that are taking over the population as the selective pressures change during, a pandemic. It's super fascinating.
All right. Well, thank you very much, guys. We said that we would go for about a half an hour on questions, and we have since done so. So if you did have a question and you didn't get it in, I'm sorry, but we've got to cut it off at some point. So I'm going to give quick channel updates, and then I will give Sunflower a chance to say goodbye, then Dr. Dan a chance to say goodbye, and then we will have our end credits, and we will be out of here. So let's see. Coming up on Tuesday is Kent with Bent. I don't know the number. I don't know the title, but it's going to happen. We're doing another Kent with Bent. Thursday is going to be the finale of the Helping Cindy Lincoln series. At least for now. She and I are still talking, and if I feel like there's other stuff that she's put out that I want to address, it might end up extending that series indefinitely. Who knows? It'll be like The Simpsons. It should have been canceled years ago, and it still hasn't been. Then, let's see. Saturday, I don't actually know what I'm doing on Saturday. However, if Answers in Genesis decides to copyright claim yet another video, maybe I'll do another stream about that. So that's about as far ahead as I want to look. I could go farther, but, gotta keep it at least a little bit of a surprise. I think that's it for the channel. So I do hope to see all of you here on Tuesday. And Sunflower, any final thoughts or, any place people might be able to catch you online or anything like that?
Final thoughts. All it would be was thank you to both of you, Dapper Dan for hosting, Dr. Dan. Thank you for honestly, just coming and having such a informed perspective and being able to convey stuff in a really digestible way. Even if this was like a private stream that no one else ever saw, I'd be like, well, I at least learned something personally. So this was just, I see it as just a great conversation, discussion. Aside from that, no, I don't really, I generally, I pop into debates now and then, but I don't really produce content on my own, but I'm more of a participator. Anyway, that's about all I got. Thank you guys so much. I really hope anyone watching enjoyed it too and learned something. And that's about it. Thanks again.
All right, thank you. And Dr. Dan, you got anything?
Yeah, I don't have anything specifically scheduled because I don't have a schedule on my channel. Every now and then I do a thing. So I've got a couple things that I want to do that I have. Relax. Now he's getting loud. Just relax. We're almost done. Why are you being loud?
Donkey noises.
Quiet. Just chill. Just chill. I mean, he's adorable, but he's being loud. So I have a couple things. I want to do a thing on junk DNA. There was a Twitter thread of all places that I was participating in last week at some point. Bean, will you just
Twitter, of course, is the height of intellectual and academic discourse.
That's where all the intellectual discourse happens. It's on Twitter, right? So but it was about junk DNA. So I want to do bad creationist genetics on that at some point. Those are typically live streams rather than pre-recorded videos. I don't have a schedule on that, but my semester is winding down, so I might have time at some point in the next couple of weeks. We're gonna see. Yeah, that's what I got. I have a couple other things planned, but I have to have time to record and edit the videos and stuff. So it's eventually I'll have some more things. Check out my channel. It's called Creation Myths, and you'll see when I do things. And Sunflower, thank you so much for the kind words and for doing this. I had a ton of fun. This was fun, so thank you.
And I do want to point out that Dr. Dan's channel, Creation Myths, is tagged in the video name. So you can click on that tag and it should bring you to his channel. If it doesn't, then I don't know how YouTube works and you guys should help me with that. So I think that is it for us. Thank you very much to both of my guests. Thank you to my audience. And as my audience has pointed out several times, and I agree with, this has been one of the most pleasant and civil discussions that we've had on this channel. It's not often that a creation evolution debate goes this smoothly. So I want to say thank you once again. And we are out of here, guys. I just want to take a moment to thank my channel members and my patrons on Patreon. They really help keep the lights on here. And I want to thank those pledging $20 or above, especially Bob Knob, Bent Hovend, Ian Chen, Chris Love, Sphincter of Doom, The Venerable Weed, and Patrick Dennis. Without the support of those whose names you see on screen right now, the channel simply wouldn't exist as it does now, and I certainly wouldn't be able to do 2 to 3 videos a week for over a year and a half now. If you would like to help support and make it so that this channel is something that can keep going, there is a link to join below the video, as well as a link to the Patreon in the channel description. If pledging money isn't right for you, please just hit like on this video, subscribe to the channel, and share my videos. That really helps my channel grow.